Assalamualaikum warahmatullahi wabarakatuh. My name is Andi Fatima Bahar. I'm from Faculty of Agriculture, University of Hasanuddin. I will present our research. The title is Isolation and Antagonism of Chitinolytic Bacteria from Ipomea caprae against Lasiodiplodia pseudotheobrae. One type of botrytis fungi that has been reported to infect crops is Lasiodiplodia pseudotheobrae, which causes grape stem disease, Democarpus logan fruit rot disease, and the last reported that Lasiodiplodia also can infect several diseases in cocoa cultivation. De Silva 2019 has informed that pathogenic Lasiodiplodia species cause blight, stem cancer, fruit rot, diabetic grape stem disease, and mimosis. Many articles have reported that chitin is the main component of fungal cell walls. It is reported that chitinolytic bacteria can produce chitinase enzyme, which can impede the growth of fungi. Several types of rhizobacteria, such as Bacillus subtilis, which can not only play a role in increasing plant growth, can also protect plants from infection with Fusarium oxysporum. Most of the chitinolytic activity is present in several species, such as Streptomyces, Serratia vibrio, and Bacillus, and also the cell walls of various types of fungi. Based on the above backgrounds, it is necessary to carry out further analysis regarding the potential of chitinolytic bacteria isolated from Ipomea caprae root to inhibiting the growth of Lasiodiplodia pseudotheobrae. There are three steps in this method. First is colloidal chitin preparation. Colloidal chitin was prepared following a modified method VDS-2T 2007, providing as much as 10 grams of chitin powder. The second is isolation of chitinolytic bacteria. The samples taken from the coast of Tanjung Bayang, Makassar, 5-gram samples were at 10 milliliters of sterile distilled water, crushed the samples and made series of dilution and spread on colloidal chitin agar. The last is interaction of chitinolytic bacteria and Lasiodiplodia pseudotheobrae in dual culture method. The chitinolytic bacteria were scratched on the surface of nutrient agar and potato dextrose agar media, then incubated for 48 hours. The results of the isolation showed that 4 bacterial solid had chitinolytic activity. Bacteria that produce chitinolytic enzyme are characterized by the presence of a clear zone in the colony area. We can look the figure 1, isolation of chitinolytic bacteria for 22 hours. The presence of chitinolytic bacteria and antagonism against Lasiodiplodia fungi on potato dextrose agar. We can look the presence in table at the table and figure 2. The presence of chitinolytic bacteria antagonism against Lasiodiplodia fungi on nutrient agar. We can look the percent at the table. Chitinase is an extracellular enzyme that plays an important role in hydrolyzing chitinase. The physiological function of chitinase depends on the source. In the plants, chitinase is generally induced by stress factors such as infection with chitin-containing pathogens. According to Gamiri et al. 2015, fungal cells generally consist of chitin, glucans, mannans, and glycoproteins. In the dual culture method experiment, the antagonism of bacteria the fungus on potato dextrose agar media did not show any significant inhibition. This is because the condition of the medium is not suitable because it contains high glucose for bacterial growth. Hasana Ilmi and Mangunwardoyo 2009 have conducted an experiment on the effect of medium containing glucose, which in the experiment showed that test bacteria did not produce chitinolytic enzyme. According to Mod et al. 2003, glucose is the most efficient carbon source for bacteria, so that the isolate grown on the substrate use glucose for growth and metabolism. This results in the production of other carbohydrate degrading enzymes such as chitinolytic enzyme, which are not reduced. According to Mod et al. 2003, this is called catabolite repression. Catabolite repression is a process by which gluconeogenesis is inhibited when glucose or other carbohydrate carbon sources are available. This is also supported by the point opinion Laley et al. 2007, which states that when sugars consisting of different monomers are exposed to acid or enzyme activity, the simple monomers are released into the medium, which are then used for physiological needs. The antagonism mechanism in the 4 bacterial isolate is likely to act as highly active microparasites. Microparasitic consists of 4 stages, namely chemotropic growth leading to a chemical stimulus, the introduction of specific chitinolytic bacterial strains, and host plant pathogens, attachment and binding of bacteria to target pathogenic hyphae, and the last process is degradation of the pathogenic cell wall. The conclusion of this research is the isolation of chitinolytic bacteria on the roots of Ipomea caprae result in 4 isolate coded, namely IPAR1, IPAR4, IPPA2, and IPPA3. The best isolate codes to inhibit the growth of Lasiodiplodia pseudotheobrae based on the dual culture test were IPPA2 and IPPA3. That's all from me. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.